Okay, class. So this is from the lecture 19 slides, which is eukaryotic gene regulation. And we're going to call this lesson 24. Um, and I'm not sure how far I'm going to get into this. I'll probably lecture for about an hour and 15 minutes. Um, and then this will get split into two, or uh, I'll get through a majority of this, and then we'll continue on to the next lecture, which is the last lecture, which is developmental genetics. Okay, so just a reminder that a lot of the stuff we've already covered when we covered in the beginning transcription and translation, and a lot of the stuff we talked about from the previous chapter two, which when we're talking about the lac operon and the tryptophan operon. Okay. So eukaryotic genes are a little different than prokaryotes. So we just went over prokaryotes, and you know that can have multiple Shindell-Garno sequences in their transcript, and so they can translate multiple proteins off of a single mRNA transcript. Eukaryotes don't do that. Remember, they always start from that 5' cap, and then they scan until they reach the start codon, whichever the first one is from the 5' end, and then they read until they hit the stop codon, and then they stop, and that's it. So you could have multiple ribosomes, but you're only going to get that one product from that one mRNA. Now remember, upstream from that, we can have alternative splicing, but once the mRNA leaves the nucleus, whatever's read by the ribosomes there, um, I mean, it can get modified, but whatever that protein codes for is what it's going to code for. Now, there's a leader, there's gonna be an untranslated leader region, and that's where we get modifications of different sugars and lipids. So we can get lipoproteins like low density lipoprotein, which is really a lipoprotein, not cholesterol. Or we can get glycoproteins, which are sugars attached to proteins, and that makes our blood groups different between, say, A and B blood type. Isn't really the protein, it's where the sugar is attached. Okay, so we have lots of different cell types. We have complex development, which we're going to cover in the next chapter. We have a lot of variation when the gene is transcribed and how much is going to be transcribed. Remember, we talked about like there's these transcription factors, and there can be lots of them. And I'll show you later on a slide of kind of what this looks like, but it's much more complicated in eukaryotes. I have seen promoters and regions with transcription factors that and enhancers and silencers that have hundreds of these. So it's super complicated, and each one of these varies. Kind of think of it as a barcode, that UPC code that you see on your groceries. So, you can have hundreds of those bars, and that's going to distinguish how your mRNA is going to be transcribed. How much is going to be transcribed, how little is going to be transcribed, if it's turned on or off at all. Um, and remember, we've already talked about this, but all of the DNA is the same in all of your cells, right? You came from a single fertilized egg, a zygote, and everything that comes from that has the exact same genetic code. What makes one cell different from another cell is how the genes are expressed. Um, so your lung cells have the exact same code as your brain cells. The reason that they're lung cells is because they have different genes turned on. The reason that they're brain cells is because they have different genes turned on. Just like a television, right? So your television it's mostly red, blue, and green. There are some fancier TVs that have magenta or some colors in it. But for the most part, it's three colors. That doesn't mean you can only watch red, blue, and green. Um, those are displayed in pixels in different areas as different colors. So together, you can watch an infinite number of pictures on your television with just three colors. Okay, so given that most genes aren't expressed in a given cell um, at a given time, and to do this, right, all the cells are the same. So to do this, either they're not getting signals from the environment, or they've shut down genes by remodeling the chromatin. So remember, your DNA is like, think about it as an ancient scroll, right? You roll it out, you can read it, and then you roll it together, and you can't read what it says at all. Your DNA is wound around those histones, like we have already talked about. And so, if it's tightly wound around those histones, it's like a scroll that's been shut, and you can't read it. So the genes in a condensed form are turned off. They could be perfectly good genes. They could be genes that are supposed to make your hair brown. But if the scroll is shut, the genes are never expressed. You're never going to make a brown pigment, and so by default, you're going to have blonde hair. This happens a lot. We have chromosome remodeling a lot based on hormones and things like that. Uh, a lot of pregnant women, their hair color can change and things like that. And it's not because their genes have changed, it's because their chromatin is becoming remodeled based on the environmental conditions they're being exposed to. And in this case, the fetus itself. Um, so there isn't like a rule of functional organization because we have a lot of things like we talked about in the previous chapter that are trans factors, proteins that can be made on chromosome number 1 that control chromosome number 16. You can have hormones that are made in a completely different region of the body that affect, like what a hormone that's made in your brain that affects your kidneys. And if you had anatomy and physiology, you know the physiology behind it. But there's not really a rule that I can lay down that says this is going to happen. All right. Let's see. So if we look at bacteria, they don't have histones, so they can't do chromosomal remodeling. Remember, we looked at bacteria, and bacteria have, these activators, these transcription factors that bind. We talked about these, the alpha and beta and sigma subunits way back when. And so if you forgot this, you might want to revisit that um, lecture. So that would be um, when we looked at RNA transcription and processing, which would be lecture 3. And then a little bit of protein and protein synthesis in lecture 4. Okay, so if we don't have the activators, right, then this isn't going to be expressed. And we can have a repressor that bounds there, like we talked about uh, in the lac and the tryptophan operon. So there's the operator region, and it can turn things on and off. In eukaryotes, most genes are turned off. And then we have things that uncoil these, that like transcription factors. But we can also have very distant things. That's what this line represents. Very distant things called enhancers. These are DNA sequences, so they're cis elements, right? Cis elements are DNA sequences that the trans elements, the proteins, recognize. And so we have all of these different proteins recognizing these DNA sequences uh, in the promoter, which we've already talked about. And then now we're kind of talking about enhancers that can be thousands, tens of thousands of nucleotides away from the gene that can control it. So it's kind of unusual. Um, in bacteria, all this goes along locally. Um, And then here we have the top box we talked about. And the RNA polymerase binds to and starts transcription when all these factors are present. Okay, so in eukaryotes, the DNA is isolated in the nucleus. That's different than prokaryotes. Transcription occurs in the nucleus. That's different than prokaryotes. Transcription and translation occur in the cytoplasm together. We have the RNA processing we've already talked about. The Once the cap and the tail and the alternative splicing have occurred, the mRNA is exported out of the nucleus into the cytoplasm, and it's read by the ribosomes into amino acid chains that we call proteins, or polypeptides is another name for it. So we can control the expression of these genes at multiple levels. We could control it at transcription. So if the transcription factors aren't there, so for example, um, in genes that are regulated for estrogen. So I study breast cancer, and that's kind of my jam. I did that for many years at the National Cancer Institute. And if we look, there's definitely a correlation between estrogen and breast cancer, and so we're looking at genes that respond to estrogen. So estrogen, um, there is an estrogen response element. So that is a cis element, right? It's a piece of DNA that's a sequence that's found in genes that are turned on or off in response to estrogen. Estrogen enters the cell. Um, there is a receptor in the cytoplasm that it binds to, and we'll talk about this later. But anyway, estrogen itself binds to and turns on these genes. So if estrogen isn't present, then transcription doesn't occur. So you could control the genes at a transcriptional level by whether or not these transcription factors are present, if the hormones are present, or whatever. It can be turned on or off, or transcribed at many different rates, right? A lot of mRNAs, a little RNAs, and in the end, you're going to get a very vast difference in the amount of protein that's produced. We can control it in processing. So we know about RNA processing. We can do alternative splicing. We can make different genes. Uh, the transport of RNA. So we didn't talk about this, but there are regions that between the start and the stop codon that are transcribed and never translated, and those tell the RNA where to go. So there are, and hopefully you remember this from Bio 181, but there are free there are free ribosomes and there are bound ribosomes. The bound ribosomes are bound to the endoplasmic reticulum. So these are usually proteins that are going to be modified, like we talked about with sugars or fats, um, or there's others. Free ribosomes are usually not modified, and free ribosomes are uh, generally things that are going to stay in the cell. So there are signals on this RNA between. So let's say that this is the start, which is AUG, and then let's say this is stop. So this is UAA. So this is what we call an untranslated leader region. This has a signal in the DNA that tells it where to go. Should it go to the free ribosomes, or should it go to the bound ribosomes? Should it get modified or not? So there's a lot of signals in here that we're not talking about, but that is what we're talking about by transportation. Degradation, right? So we talked a little bit about how we have RNA interference, and we have lots of ways to degrade RNAs once they're made. Um, We can control it at the translational level. So we can block uh, the ribosome. We can interfere with the ribosome. There's lots of things going on here that we didn't talk about. And if you're interested in this stuff, you can take a class called molecular genetics that I teach occasionally at ASU. And that class talks a lot about this kind of this level of control, which we're not really covering in detail. And I don't expect you to know that. I'm just kind of showing you we can control it at the processing, so whether or not it gets modified with sugars or fats, or the protein gets cut in half, or we have a phosphate to it. And then there's a whole thing on protein degradation, um, how the proteins are broken down in the lysosome or other ways. So we can control all of these levels of gene control to, in the end, modify how much protein is being produced by these. Okay, if we look at transcriptional control, which is what we're mainly going to focus on, um, we're going to get signals from the environment, like we talked about. So, it could be uh, estrogen, or, I mean, it may be just cold outside. And so you're going to respond to that cold by changing the cholesterol levels in your genes and all kinds of other stuff. And that's going to affect the transcription rates. We can either turn it on or off, or we can turn it on a little or a lot. Um, so we talked about protein factors. We talked about transcription factors that activate or inhibit transcription. So we have transcription factors that are activators, or transcription factors that are repressors. Um, and then I just talked about this. So in eukaryotes, um, we can have these sites. They're called enhancers and silencers, and these can be tens of thousands of nucleotides away from the actual gene. Some regulation is epigenetic. So I'm not going to get really crazy into epigenetics, but this is a branch of genetics so epi means above. This is a branch of genetics that studies traits that may be inherited but not expressed. So if you look at twins, right, they're genetically identical. But one twin might get cancer and the other twin might be cancer free. So what caused the cancer, right? Is it an environmental factor? What is causing that? And so if you study epigenetics, you're kind of looking at, like, how does that happen? And it may not even be a mutation in the DNA, right? So you would think, okay, well, that twin got cancer because they smoked a lot of cigarettes or whatever, but they live similar lifestyles together. It's not like one lives in a uranium factory and the other one uh, lives in a lead-lined box. So um, what scientists have found is, and we're going to talk about this, is that methylation of histones, shoot, or acetylation, and again, we're going to talk about this, of histones, can cause the chromosomes to remodel, which means basically that the DNA, the scroll of the DNA is open or closed, right? So let's say two twins inherit a perfectly normal brown gene, which I've already said, and one is shut down. It could be just because one parent smoked, right? So it could be their gene, and that gene it's perfectly good. It's just closed because of chromatin remodeling. So it can't be read. A good gene, even though it's perfectly intact, there's no mutations in it or whatever, if it's not transcribed and translated, it's going to look like it's not functional. And that's what epigenetics is. Um, Neil deGrasse Tyson did a whole, like, he's kind of into epigenetics and astrophysics. It's kind of me in backwards because I dabble in astrophysics, but it's not like my main gig. And he dabbles in biology and genetics, but that's not his main gig. His main gig is to destroy my childhood teachings of Pluto being a planet. Anyway, so uh, yeah, that's all I'm going to say about that. So if you looked up Nova and epigenetics and Neil deGrasse Tyson, you would find it, the video on it. Basically, it talks about twins and how your diet and what your parents do or whatever can affect your genetics, whether the genes are turned on or off. All right, so this should be a review, but we have regulatory proteins. Remember, these are the trans elements. They can move around in the nucleus or the cytoplasm if you're their bacteria. We're talking about the nucleus here. And then there are regulatory elements. So these are the DNA. So these are the cis elements, right? They never move. Trans elements can move. Trans means across. Here's the promoter, the top box. That's a cis element, right? RNA polymerase is a trans element. And all of these are required to get the RNA transcribed. So no big deal. You've seen this before. Eukaryotes don't have operons. We talked about it. Uh, so in operon structures, all of the gene products are made generally in equal amounts because you have one mRNA, right? So the ribosomes are going to make, let's say they make 10 units per mRNA. So they're going to make 10 units of LAC Z and 10 units of LAC Y and 10 units of LAC A. That doesn't happen, but there is coordinately regulated genes that can be dispersed throughout the cell. They generally share what we call promoter proximal. So promoter proximal things are things within the promoter region. We talked about these. So, you know, the cat box and the GC box and the, you know, things that are close to the promoter or within the promoter are called promoter proximal. And then there are promoter distal, which are distantly found elements, DNA cis elements, that can be what we call enhancers or silencers. There is very few, I mean, I had a student once, I don't know why, they were just bored and they researched this. So I said, there's no eukaryotes that use an operon structure. So you can't make multiple proteins off the same mRNA. And of course, once you say that, like, there's millions of species, then there's going to be an exception. And so one student found some fungi that was from Brazil that had an example of a single mRNA that could be read in different ways. And I'm not sure, they showed me the paper and I read the paper. I'm not sure exactly how that was regulated in a eukaryote, but so I'm going to say that there's none, but of course then I would be proved wrong. So as far as I know, there's one instance of an mRNA that can make multiple proteins. All right, so we talked about this. Cis-adenine sequences, these are DNA sequences. They don't move. They're in the core of the promoter, like the top box. They're near the promoter, like the cat box or the GC box that we talked about. And then we have things that are far away. So these are DNA sequences. They're cis. We call those enhancers. There are things that are far away. We call those silencers. So this is the region we have in the promoter, close to the promoter, far away from the promoter. And then these transacting controls are done by, we talked about transcription factors already, that these can be genes that are linked, right, on the same chromosome or unlinked, because transcription factors are proteins, and they can move around the cell no problem. They're just diffusing in the nucleus or the cytoplasm. Okay, so these are some terms that you have to know, and so just get you a note card out or whatever you want to do, however you want to memorize this stuff. But we have positive and negative regulation. So when we're talking about transcriptional regulation, so positive regulation, we have an activator that causes transcription to occur. The activator binds to the operator. This is kind of like the cyclic AMP cap protein, right? It's positive regulation. We need the cyclic AMP and cap protein to be present to cause transcription to occur. And in general, if that's not present, then we don't get transcription. Negative regulation is we have a repressor that's bound, and when that repressor is bound, it blocks the polymerase so we don't get transcription. And if the repressor is absent, then we get transcription. So that's negative regulation, that's positive regulation. All right, so we've already talked about these, and this should be a review. We have Tata, which would be core sequence. We have the cat box, around 100 base pairs. We talked about this, 70 to 200 base pairs. This one, GC box, 70 to 200 base pairs. We would call these core promoter, distal, proximal promoter elements. And we've already covered these, so this is just a review. I'll just go through this pretty fast. So if we mutate these sequences, right, they're core to these transcription factors that affect transcription. So you can see this is just a DNA sequence. This is the start site. So this would be, I don't know if it'll let me write on the slide. This would be +1. This would be -1. This is around -35. This is somewhere around 70 to 200. -70 to -200. Same thing here. And you could have, these could be changed in all over the place, but this is just an example. We mutate this, we get decreases in transcription. Right, we mutate this, we get decreases in transcription. We mutate this, and that makes sense because these things help regulate transcription. So if the sequence is not correct, the transcription factor can't bind and activate transcription. So let's see. All right, slide 11. Okay, so remember, cis sequences are DNA sequences, and if they're far away, we call them enhancers or silencers. So they can act at a distance from the promoter, and this is doable because DNA isn't like a still piece of bar. It is movable. It's more like a shoelace. So you could take one end of your shoelace and fold it over to touch the other end of the shoelace. No problem. And that's how DNA works, right? It's not a stiff piece of steel. It moves. Let's see. Okay, sorry. So that was my kid's school, and apparently he rolled his ankle, so they're concerned about it. But I don't think it's that serious, so I'm gonna finish this lecture and then I'll go check it out. Okay, so we were talking about enhancers and silencers. So enhancers, and we're talking about DNA, how DNA can fold, right? So these enhancers can be thousands, tens of thousands, hundreds of thousands of nucleotides away because the enhancer will fold. So we have the enhancer and interact with the promoter core or promoter proximal elements, right? So we can have trans elements that bind to these enhancer sequences and then affect transcription. Either turn it on, and then we usually call that an enhancer because it binds activators. And if it turns it off, we usually call those silencers, and those bind the trans element repressors. So activators and repressors are proteins, and these are trans, right? They can move around the cell. Enhancers and silencers are cis elements. That means they're DNA sequences, and they are fixed in the DNA. We can fold the DNA, right? But they can't. This can't jump out and come over here. We're not talking about that. I mean, it could, but that's a whole nother deal. Like that has to do with transposons, and we're not going to talk about that this semester. Anyway, so these are tissue specific, and they can be stage specific, meaning that what stage of development. Okay, so I don't care that you know this. I'm not asking you to memorize the LEF1. This is just an example. So we can have an enhancer protein. So let's say that these two regulatory elements are far away, and then we have something that bends the DNA, and brings them together. This is like the arabinose operon that we skipped. Um, so the reason I'm showing you this is because I want you to see that two very distant things of DNA can interact with one another. Hang on. Okay, sorry, my phone's blowing up. Anyway, so what I want you to get from this slide is that DNA is mobile, it can move and bend, it's not rigid. Good enough. All right. One other thing I forgot to mention is these are orientation independent. That means that the sequence can be in one direction or another. It doesn't matter, right? Because it's folding. So you could have something bind onto one strand, not the other, or on the other strand and not. And so anyway, it doesn't matter if this could be forwards or backwards. It can be flipped around. It could be upstream of the promoter. It could be downstream. Doesn't matter, right? These are completely independent. Orientation independent and position independent. All right. So, all right, here's an example of an enhancer, an activator. So here's the enhancer. Remember, the enhancer is a DNA sequence. It's this sequence. Activators are proteins. Right. So these are trans elements, proteins. These are cis elements. They're DNA. So these activators recognize these DNA sequence and bind to them. Right. Here's our promoter. Here's a promoter proximal or promoter core elements, the top box. And so something could cause this DNA to move, like maybe another protein, or it could just be that these have an affinity for one another. So here's all the transcription factors that are assembling, including our enhancers that have already bound their activators. And all these things come into place, and maybe they're interacting with a transcription factor here to allow the appropriate shape, to allow the polymerase to bind to start transcription. So these can be very far away, these enhancers, from the promoter itself. All right, that's enough of that. This is just another example. I'm not asking you to memorize any of these transcription factors, but remember we talked about these A, B, E, and D way back when. And then we have the enhancers and the activators, which are interacting with all of these transcription factor elements and the polymerase to activate transcription. Okay, so there are repressor proteins that can reduce transcription, either directly competing or quenching. So what's the difference? Okay, so let's go through this. All right, so here's an activator, right? It's a protein. Here's a repressor. It's also a protein. Here's an enhancer. All right, so remember, the activator binds it. We call it enhancer. The repressor binds it. We generally call it a silencer. But they can be competing for the same spot. And this is kind of an example of this is that lambda phage that we didn't really cover. But if there is more, let's say there's more repressor than activator, then the repressor will outcompete the activator. And maybe there's more activator than repressor, and the activator can outcompete the repressor. Anyway, regardless, if one binds, the other one obviously can't. If someone parked in your employee of the month spot, even though you're the only employee of the month, you can't park there. Um, and so let's just say the gene here, uh, this is the protein. So it's if the repressor binds, then it's going to repress the protein being made. If the activator binds, then the protein gets made. Okay, so so that's competitive binding between activators and repressors. We can also have something called quenching. So an activator can have has a DNA binding domain, right? So they also, and we talked about this. So we talked about that proteins can have multiple. This is just a square protein, right? It could have a DNA binding region. It could have a transcriptional factor activating region. It could have a hormone binding region. I don't know why my handwriting or this pad is so terrible today, but anyway, hormone binding. So this site could be binds to hormones, right? Like maybe estrogen. And this site could bind to the estrogen response element, which is a cis factor, a DNA sequence. And then maybe this interacts with the polymerase to turn the gene on. So you can have one protein with lots of different roles. And so we have this one. One side is activator, one side is the DNA binding. Here's the repressor, right? So in some instances, the repressor can bind to the DNA binding domain. If it binds to that, obviously the activator can't bind to the DNA because it can't recognize it. So in that case, uh, it's blocked, and we don't get the protein being produced. Type 2 is the repressor can bind to a block the activating domain. So maybe the DNA binding domain is fine and it binds, which is shown here, to the cis element. But the activating domain is blocked by the repressor. And in this case, it can't interact with the polymerase, and so we get no gene expression either. So that's quenching. There's two types of quenching, and so it can block any of these domains and quench it. Okay, so here's the arabinose. We didn't cover this. Just kind of ran out of time, but anyway, remember we have transcription factors that activate. This is the TATA binding protein. It's obviously going to bind to the TATA. We can have inhibitors that bind to that too. We talked about all of these transcription factors that interact with it. I'm not asking you to memorize them. Um, so we can have a combination of these cis factors binding multiple transcription factors, and that kind of acts like a dimmer switch. So it can turn the gene on or off or anywhere in between. And that those elements, those DNA sequences, regulate gene expression. Together, um, I mean, we could read through this. It's like you could combine these things so that you have different arrays of transcription factors bound to them. Uh, you can have positively regulated proteins that bind to an enhancer and a promoter and activate transcription. You could have repressors bound, and it can interfere with the activation of the promoter and gene expression. So the complex of all these things is called an enhanceosome. Um, and these things can get really complicated. So you should know this term. It's probably going to come up again in some class. All right, so I'm not asking you to memorize this or whatever. I'm just telling you that you can have a combinatorial control over this. This is, and we're going to talk about this in developmental genetics, but this is how a single cell, a single zygote, can become multiple different cells uh, through external cues, environmental cues, regulations of genes that are inherited, and things like that. So let's say we have a precursor cell. There's some sort of regulatory protein that so we talked about estrogen, but this could be something like, I don't know, insulin that binds and causes this cell to differentiate. We'll talk about lots of these different proteins. So, I mean, insulin, I guess, is a terrible one. We can say antennapedia. We talked about that one, right? So, antennapedia gets activated by some external thing. We're going to talk about bicoid and nanos. You never heard of these yet, so I don't want to use that as an example. But, antennapedia is expressed, and that makes legs. And it's supposed to be repressed in the head so that the antenna of the organism doesn't become legs. So that repressor isn't working properly. It's not getting repressed. So that gene is expressed and it makes a leg instead of an antenna. And we've already talked about this. So let's say that happens. Then we get a different cell line. Another regulatory protein comes along. And so we can have a combination of all these different genes turned on to make different cell lines, right? Like a lung cell, a heart cell, a brain cell, a skin cell, whatever. All right. All right. So this is just the beta subunit of hemoglobin. This is showing you that there's lots of things that are involved in regulation of this gene. So these are generally promoter proximal or promoter core promoter elements. Here's the Tata box. This is minus 30. So Tata is generally minus 35. You learned that. And I'm not asking you to memorize these things, right? I mean, GATA stands for GATA, which is the sequence that it recognizes. Some of these things have some weird names. The artist that drew this just made the proteins into fruits, which is fine. So it's kind of funny. You could have this apple and this lemon competing for that site, right? We talked about competition. This carrot at different protein vines. Here's another GATA site. Here's another lemon, and so on. I don't even know what that is. This is a squash. So anyway, here's a banana. These are competing for sites. So these elements can be within the promoter, and then these are at the end of the gene, right? This is regulates the gene as well, because DNA can fold, right? So anyway, the reason I'm showing you this is because I want you to understand that there are lots of things binding to lots of trans elements, in this case the fruit, binding to DNA sequences, the cis elements, that are within the promoter. And then this is just showing you, this is, even though the artist didn't draw it to scale, this is several thousand, so 2200 to 2400 nucleotides downstream of the promoter itself. All right. Here's the enhanceosome, so it's just a combination of all this stuff going on in there. Here's another image of it, the eukaryotic enhanceosome. So I'm not asking you to memorize all of the things that go into this, but you can have multiple silencers, enhancers. You can have multiple cis elements that are, co-activators, transcription factors. We talked about repressors, activators that are binding to silencers, enhancers. So this can get really complicated really fast. All right. So that's kind of an overview of how eukaryotic organisms regulate their genes, and we're a lot more complicated. So we have the same DNA, but we have to make multiple cells from it. So we have to regulate it differently. Um, these okay, so the regulatory proteins. This is just a review. Uh, they're made from mRNA and then they're read by ribosomes into a protein. So they're a product of transcription and translation. They have lots of domains. We just talked about that. So you could have a protein. That's my square. So this could be the DNA binding, binds to the DNA. This is a transcription factor that turns on the gene. This is uh, let's see. It could bind to it like a regulatory protein, another regulatory protein, or even say like a hormone, like the estrogen response element. And then this might, bind a phosphate group that regulates it. So if the phosphate binds, maybe it turns it on, and we take off the phosphate, maybe it turns it on off. Um, so environmental sensor conditions. So that things can change their shape due to temperature. Like that's why they put expansion joints in bridges. So if it gets hot, it expands. If it gets cold, it contracts. Your cell is no different. In fact, your cholesterol level is regulated based on the temperature that's outside in your cell. Um, all right, so here, this is just another drawing of what I've drawn out twice now. So beating the dead horse. Okay, so we're gonna go through the different kinds of things that can bind to DNA and regulate it. So these are proteins. They have, remember, proteins are made of amino acids, so they have an N terminus, an amino terminus, and a carboxyl terminus. Um, and then we talked about alpha helices and loops, and we talked about beta sheets. So when we're talking about a helix loop helix, that's an alpha helix with a short piece of DNA. So it's not drawn here, but I can fill it in. It's just a short piece of DNA that connects the two. It's not long enough to actually be a beta sheet. So this is a dimer, right? These are two, and these are nonpolar contacts. I don't care that you know that, but you should know that there's a helix loop helix motif, and this is how geneticists talk. These helices fit into the major groove of DNA. So remember, DNA has a major groove and a minor groove. So this would be the minor groove. This would be the major groove. Um, an example of the helix loop helix. Remember, these are trans regulatory elements because they're proteins. And so they fit into the major groove because it's bigger to look at the DNA to test the sequences to see if they match. And then they bind to it. That's the DNA binding specificity of these trans regulatory proteins, right? So we're talking about this thing right here. All right. So examples of this. So I think we talked a little bit about this. We're talking about cell cycle, but an oncogene is a gene that turns on the cell cycle. Proto means that it hasn't been mutated to be out of control yet. So there's a oncogene. Once it gets mutated, it becomes an oncogene called c-Myc, and that leads to cancer. So knowing how these things function is important. It can be important in cancer and stopping cancer. There are helix, turn helix. So this we have three domains. This is showing it binding to the DNA. Three helices. Helix 3 binds to the DNA. Helix 1 and 2 bind to other proteins that regulate gene expression. So this one is the important one, right? It's interacting with the DNA sequence itself. So this is the DNA binding domain. And then these are the transcription factor, transcriptional regulation domains. So an example of this is the lac repressor, right? We talked about the lac repressor, and in this case, it simply binds to and blocks the polymerase. And we talked about the tryptophan repressor as well, so we've covered those. CRO is from lambda phage, which we didn't cover. And then homeotic proteins are ones that are involved in development, which we will cover in the next chapter. Or the next lecture, which that will be lecture 20 when we talk about developmental genetics. So I'm not going to ask you about this, but I want you to know what a helix turn helix is, right? I want you to know that this thing binds to and interacts with a major groove of DNA and regulates transcription. So turns it on, turns it off. There are a thing called zinc fingers. So zinc fingers, they're called fingers because they look like fingers and they bind zinc, right? So it's showing right here that it's bound zinc. It can be a monomer or dimer. Zinc is generally bound between two cysteines. So this is kind of, just to try to give you a real world example of why you might want to know this. So there are drugs, and I'm sure that you've seen them on TVs, like antibodies. And so, let's say we inject an antigen. So antigen means antibody generator. That's what it's. So these are just proteins that are foreign to the rabbit or the cow or the pig or whatever you're growing it in, and you put it in there. So let's say you want to make antibodies to, I don't know, let's say COVID, right? Just for the lack of any like a better thing, but you see these drugs all the time. If it says AB on the end of the name, it's an antibody. So let's say you take COVID, you inject it into a horse. They do this with snake venom, whatever, and that animal, this is foreign to it. It's going to make antibodies to it. So the question is, how do you purify this so that you can give it to the person that's bit by the snake or the person that has COVID that, to help them fight whatever that is. And so what they do is they pour it through a column. They if you took a biotech class or a genetics thing, there's easy ways to isolate proteins. We can isolate the proteins from all the other stuff in the cell, the lipids and the DNA. And so if we isolate all these proteins, we pour them through a column, right? There's things on the side of this column that bind. And so a lot of times when we artificially manufacture these proteins, or these antibodies, we add like a multiple cysteine chain on it to make a zinc finger, and that or histidine could be like histidines or cysteines, both bind to zinc, and so you line the column with zinc, and when you pour this through, these zinc fingers bind to the zinc. So everything that you don't want goes through the column, and everything that you do want is bound using these cysteine and histidine residues to the column. And then to get them off, you can just modify them chemically. So most of the time you just change the pH. Things that change shape, right? Things that change the shape of proteins very quickly are pH and temperature. So you could change the temperature to release these proteins, or you could more commonly just change the pH. So once you get rid of all this stuff, throw that away. You change the pH, and then you run a buffer through here, and then in the end you end up with your product, right? Your vaccine to COVID, or your shingles, that whatever, it could be anything, any drug. I've seen all kinds of new drugs that are antibody drugs that do stuff I've never even heard of. So if you watch television, I'm guarantee you've seen a drug that's manufactured like this, a protein drug. And most injectables are proteins, right? Because your stomach would break it down. All right. So anyway, that's why zinc fingers are important, right? So usually cysteines or histidines make a finger, and those bind to zinc. So steroid receptors, we're going to talk about estrogen receptor. It could be testosterone, any steroid receptor, and then we're going to talk about gal4p in a minute. Not in a minute, maybe next lecture. We might get to it. I don't know. So this is what zinc finger. I don't care that you know this. There's lots of different versions of this motif, but in the end, what you should know is that histidine or cysteine residues bind zinc, and that makes a finger, right? It's kind of giving you the finger. All right. So examples: transcriptional activators. 1% of all mammalian genes code for zinc fingers. So these are quite important. Oops. So it's just a different zinc finger motif. I don't care that you know this. This example is gal4. We're going to talk about that too. So you'll know how that works. And then we have leucine zippers, right? And so these are homodimers or heterodimers. They are formed through interaction of leucine-rich domains. Again, some of these, so these C are proto-oncogenes. Once they've mutated into cancerous genes, they're called oncogenes. So these leucine-rich domains recognize and bind to the DNA, and so this is how it's held together. And honestly, I don't really care that you know the specifics, but I'm just kind of giving you how a protein can bind to DNA. So I might ask you, what are the ways that proteins can bind? Helix turn helix, zinc finger, leucine zipper, and then for leucine zipper, I might ask, or zinc finger, I might ask you, well, what chemical element does it recognize? And you would tell me zinc. That's why it's called a zinc finger. And then this is how these are connect with one another. So these are the DNA binding, and then the leucines bind these two together. All right. So how do we control transcription? Now we know that there are trans proteins, the proteins that bind to the cis elements, the DNA regulatory elements. When we talk about an effector molecule, it's something that affects transcription. So we've already talked about this. The effector molecule for the lac operon is lactose. The effector for the tryptophan operon is tryptophan, right? Those things have an effect on the expression of the gene, through that interaction of the repressor or activator. And this isn't any different in eukaryotes. So not everyone has a lactose gene. It's not really in eukaryotes. It's not like well studied, and so but galactose is. And it's particularly studied in yeast because yeast are easy organisms to study. Fast generation time, like we talked about, lots of offspring. Um, so we're going to talk about galactose, and then we're going to talk about steroid hormones, right? So these are hormones that are lipids based, and then we have polypeptide hormones that are protein based. So, not sure if you learn this in your 181 class, but I teach it. So, intro biology. Um, a steroid hormone or steroid is part of the lipids that we cover as macromolecules. And examples of that would be like estrogen, testosterone, cortisone, corticosteroids, any of those lipid based. They're all synthesized out of cholesterol. They're all four ring structures. Polypeptide hormones are proteins, right? So an example of that would be insulin. So insulin is considered a hormone. Um, Insulin binds to the cell surface like many polypeptide hormones, never crosses into the cell. Steroids are different. They can cross through the cell membrane because it's a phospholipid bilayer. Hopefully, every one of you guys know that. So things that are hydrophobic can go through the membrane without a channel. And so hormones are no different. Okay, so we're going to talk about galactose metabolism in eukaryotes and gene expression. So again, unlike prokaryotes, we don't eukaryotes don't have operons, right? They're not polycistronic. They don't have on the same many genes on the same mRNA, right? And there's like one example maybe that it isn't. The gal genes, so this is Saccharomyces cerevisiae. Uh, I mean, I probably everyone knows what cerveza is, which is beer. So this is a yeast that produces beer. Um, this is what's mostly studied for eukaryotic gene regulation. So galactose, the sugar is it ends in OSE. So it's broken down by genes that are involved in well, lots of genes start with GAL. So we're going to talk about the major player genes that are involved in breaking down galactose. Just like we talked about the genes that are involved in breaking down lactose, like beta galactosidase and permease. Okay, so I mean, we're finding out there's a lot more genes involved in this, but to keep it simple, we're going to talk about four genes: Gal1, Gal2, Gal7, and Gal10. These code for enzymes that function in breaking down galactose. So Gal1 is a galactokinase. I don't know if your 181 instructor told you this or made you learn this. I mean, heck, I think I made you guys learn this. So a kinase is something that adds a phosphate. And when you add a phosphate, when you add one molecule to another molecule, it changes its shape. And if you change its shape, it may not function. I mean, think about a train track. If I change the shape of the wheel, it wouldn't roll smooth anymore. So even a small change in shape can have a big change in function. I think I just got to write slower. Um, you know, I teach this in 181. So sodium is a explosive metal. And chlorine is a deadly gas. And if you put these two together, you get table salt, which is completely harmless. So take if these are separate from one another, they would either make you blow up or die. Um, but they don't because this is a different shape than this, and it has a different function. It changes its function completely. So a galactokinase, job is to add a phosphate, and that phosphate changes. So this is the symbol for change, delta. its function. Permease, it's the exact same thing that you think about it, right? So when we lack Y, codes for permease. This is going to make the cell permeable to galactose because galactose, like most sugars, are water soluble. So they're hydrophilic, not hydrophobic. And so they can't go through the membrane without a channel. So we need to build a channel. Right? Here's our cell. We have to build a channel that allows galactose to enter. Right? These channels, integral membrane or transmembrane proteins, are different. Like there's an iron transport, and there's a one that transports water. And so here's a galactose one. It's not always going to be permeable to galactose. It's going to respond to whether or not galactose is present. There's a galactose transferase and then a galactose epimerase, and I'll show you how that works. So in the end, these genes are going to bring in galactose and then rearrange that to D. So this is the dextrose. So, I teach this in 181 too, but so when you have a central carbon with 4 different things attached to it, it makes an optical isomer, which is also called an enantiomer. And that those have a right and a left-handed form. So in science, we call the right-handed form D for dextrose. So D is right-handed. Think about your hands are different, right? You can't make one hand be the exact of the other. And if you're not sure about this, just look up enantiomers or optical isomers, and it'll explain to you why. Because of this complex nature of this central carbon with 4 attached different groups, we'll just call them R groups, they can have a left and a right-handed form. So D-glucose is the right-handed form of glucose. There is a left-handed form, an L-glucose. Right, that stands for left-handed. And then in medicine, usually it's R, which is rectus, which is right in Latin, and then this is S, which is left-handed, and that stands for sinister, because all left-handed people are sinister. I'm just kidding. My wife would kill me, and so are like half my kids are left-handed too. Okay, so anyway, that's the designation. D and L for science generally, and so that's why this is called D-glucose. And then R and S usually for medicine. These are usually compounds that are synthesized like antidepressants and things like that. Okay, so this produces D-glucose 6-phosphate. So the phosphate is added. This kinase adds a phosphate, and this is a product of glycolysis. So I make my students know this. This should be like a review. Hopefully, I don't know who your intro bio professor was, but the first step when glucose enters the cell, that glucose, there's a hexokinase, and that adds a phosphate to this glucose, and it adds it on the number 6 carbon. So it produces glucose 6 phosphate, right, which is the first step here, right? So we're going to convert galactose into this glucose 6 phosphate, so that it can go through glycolysis. and be broken down regularly. And then once it goes through glycolysis, if oxygen is present, then we produce um, pyruvate, and then that goes to the Krebs cycle and so on. And I'm not trying to teach you uh, cellular respiration right now. So anyway, this is just showing you. I don't care that you know this. There's lots of different metabolic pathways for lots of different sugars. This is the general pathway for we have glucose. So here's galactose, and then it's converted into the right-handed form of glucose 6-phosphate, and then that enters glycolysis. Don't care that you know that. Do care that you know this. So we have GAL1, that's the galactokinase. It takes galactose and adds a phosphate on the number one carbon here. And then once the one phosphate is present, then GAL7 is a transferase, so it converts um, this into this. And then epimerase GAL10 converts this to glucose, and then it goes through the process of glycolysis. So anyway, I'm not going to ask you what these specifically do, um, like I will for lac operon, but not for the gal operon. You should know that it's gal1, gal7, and gal10, and that these three genes are involved in converting galactose into a metabolic process product uh, like glucose 6 phosphate. It's loud. There's a bunch of people outside my office, like super loud. Anyway, so here's gal 10 and gal 1. This is an activating sequence, so it's a cis element. It's G because it's galactose. And then we have gal 10 that's being transcribed in the left-handed direction, gal 1 that's being transcribed in the right-handed direction, and then gal 7 that's transcribed in the left-handed direction. So these are the ones that we're talking about. And, we have a kinase, transferase, and epimerase. Okay, so again, this is in yeast, but it's the same for most eukaryotes. So the gal genes are near each other, but they don't constitute an operon. Here's another drawing of what we just showed. This is that upstream activating sequence that um, is in charge of activating both gal1 and gal10. There's another activating sequence that activates another gene called gal2, and then gal7 as well. And this is just showing you this is on, uh, well, this is on chromosome 2, this is on chromosome 12. And then just remember that transcription occurs in either direction from the UAS gal. Okay, so when we don't have galactose present, we don't need to produce the genes to break down galactose, right? So we want to, if there's no galactose, we want this to be turned off. So here's gal4. We have the activator region and the DNA binding region. And when galactose is absent, there's another product called gal ADP, Um, gal ADP is basically floating around the cell and it binds to the activator region of gal 4. So when gal 4 and gal 80 are bound to one another, the gal 4 can't activate the gene. So the genes are off, right? Gal 4 is binding to the activator part of gal 4. All right, so when galactose is present, um, gal 3 and galactose together bind to gal 80 and cause it to change its shape so that it can no longer bind to the activator region. And so gene expression is now able to be turned on. So when galactose is present, we have the expression of genes to break down galactose. When galactose is absent, then the genes are turned off. So galactose is an inducer, also known as an effector. And so the metabolic control is a lot more complicated than what we just talked about. And I don't care that you know that. I'm just showing you that there's a lot of things that we don't know what they do. All right. So I'm going to stop here and I will pick up on the hormone regulation part of this in the next lecture.